dendrite is a short, branched extension of a nerve cell along which impulses received from other cells at synapses are transmitted through the cell. An axon is a thread-like part of a neuron that transmits signals to other cells. The cell body is the nucleus and cytoplasm of a nerve cell, excluding the dendrites and axons. Synaptic vesicles store various neurotransmitters that are released at the synapse. The presynaptic neuron is a neuron that fires the neurotransmitter as a result of an action potential entering its axon terminal. The postsynaptic neuron has a signal that is transported to it by the release of a chemical neurotransmitter. Neurotransmitter receptors are activated by a neurotransmitter, and they cause ions to move through the channels.
The resting potential of neurons is formed by concentration gradients of sodium and potassium ions. There is a higher concentration of potassium ions inside the cell and a higher concentration of sodium ions outside. These resting gradients are maintained by sodium-potassium pumps using active transport to go against the concentration gradient on either side of the plasma membrane. This concentration gradient creates chemical potential energy, with the charge of a neuron at rest being negative 70 millivolts.
Voltage-gated ion channels are transmembrane proteins that form ion channels and are activated by changes in the membrane potential. These changes are caused by an increase of sodium ions inside the cell and a decrease of potassium ions.
When neurons activate, gated ion channels along the plasma membrane open in response to stimulus. This changes the permeability of the membrane and with it, the membrane potential. When depolarization occurs, gated sodium channels open first, allowing sodium ions to flow into the cell, making the membrane potential less negative. For an action potential, the charge of the neuron must exceed negative 55 millivolts.
As an action potential proceeds down the length of a neuron, the gated sodium channels behind it deactivate, while most potassium channels open, effectively bringing the membrane potential back to below resting potential. When the potassium channels close, the sodium-potassium pumps work to restore the resting concentration gradients. The period of time between the start of an action potential and the reactivation of the sodium-potassium pumps late into the repolarization phase is known as the refractory period. During this time, the concentration gradient of sodium and potassium are not yet regenerated, and a second action potential cannot be initiated. This puts a limit on how often action potentials can occur, essentially forcing them in a direction down a neuron by presenting backwards movement.
The axon are surrounded by a layer of mostly lipid tissue, which provides insulation for the axon, known as the myelin sheath. The insulation causes action potentials to move faster down the axon. However, there are gaps in the myelin sheath, known as nodes of Ranvier. Action potentials cannot be generated in these nodes. For an action potential to continue through these gaps, inward flow of sodium in one node travels through the axon to the next node, triggering an action potential there. This is called signal propagation. During an excitatory response, a neurotransmitter from a neighboring neuron travels across the synaptic cleft and binds to the gated ion channels. This makes the channels open, causing the membrane to be more permeable to both sodium and potassium, causing a minor depolarization but not quite reaching the charge required for an action potential.